In Malaysia, consumption of beef increases from the year 2007 to the year 2012. Poultry meat consumption increased from 1,048.59 tons to 1,348.63 tons the year 2007 to 2012. Due to the increase of beef and poultry meat consumption, there are strategies needed to develop by farmers and the government to increase meat production. In factor affecting poultry meat color, color is important when meat and poultry are purchased, stored, and cooked. Other and attractive, bright color is a consideration for the purchase. Myoglobin, a protein, is responsible for the majority of the red color. When it is mixed with oxygen, it becomes oxymyoglobin and produces a bright red color. Water holding capacity is simply for WHC. WHC of meat is affected by several of the factors which cause differentiation in muscles, such as species, age, and muscular function. The factors affecting the loss of weep, or drip, from uncooked meat also apply to the water holding capacity of cooked meat. Relative differences are retained on heating. In factors affecting meat juiciness, meat cooked quickly to a given internal temperature has a lower cooking loss and is juicier than that cooked slowly to the same temperature. The juiciness in cooked meat is due to the first impression of wetness produced by the rapid release of meat fluid and the stimulatory effect of fat on salivation. For your information, juiciness reaches a minimum when the pH level of the meat is about 6, and this possibly reflects the greater ability of the muscle proteins to bind water in this pH region. The ranking order shows that juiciness was greatest in the fresh, frozen meat of high ultimate pH, and somewhat less in corresponding dehydrated material, but both were considerably more juicy than the meat of low ultimate pH both before and after freeze-drying. The tenderness relationships reflect not only direct chronological changes in muscular and connective tissues but also associated effects due to the increased bulk and fatness of carcasses with age. These influence the differential effect of cooling conditions on the extent of cold shortening in specific muscles. Increasing consumer awareness of differences in tenderness in other attributes of eating quality between specific muscles is promoting developments in their marketing, and selecting the palatable muscles from joints overall have hitherto been regarded as being of lower quality. In post-slaughter, there are 4 parts. There are post-mortem glycolysis, conditioning, cooking, and processing. In processing, the rates of freezing normally used in commerce have no effect in this respect. Meat blast frozen at a rate sufficient to freeze the deepest portion of the carcass in 24 hours tends to be tougher on cooking than corresponding meat which has been chilled for the normal 2 to 3 days before freezing, possibly because the latter has had what amounts to a short conditioning period. While in conditioning, the large temperature coefficient of conditioning changes is the use of electrical stimulation to avoid cold shortening in meat which is refrigerated swiftly post-mortem. For artificial tenderizing, other than marination, plants, and enzymes, pre-slaughter injection of the live animal was proved to be the most effective method of introducing proteolytic enzymes into the meat so that they penetrate uniformly into the furthest interstices of the tissue. The last factor affecting eating quality in meat is odor and taste. Flavor is a complex sensation. It involves odor, taste, texture, temperature, and pH. Odor and taste are most difficult to define objectively. The use of electronic noses and tongues in flavor analysis has increased rapidly. But the evaluation of odor and taste still depends mainly on the taste panel. Response to odor occurs in the olfactory cells of the nasal surfaces and is conveyed from these to the brain for interpretation by the olfactory nerves. For taste, it occurs in specialized cells on the tongue, the soft palate, and the top of the gullet. In undesirable odor and taste, there is a gradual loss in flavor during storage, and this may occur even in the frozen condition, possibly due to the slow loss of highly volatile substances. These losses are unavoidable. Undesirable odor and taste may arise during the storage of meat because of microbial growth, chemical deterioration on the surface, or tainting by extraneous agents. One of the reasons is the exact nature of the off odors will depend on the types of microorganisms growing, and these, in turn, will be determined by such factors as the temperature of storage and the nature of the product, fresh, cured, comminuted.